41-year-old woman presents with occasional dyspareunia and vaginal bleeding after sexual encounter. She is in a monogamous relationship, uses oral contraception. She does not have a family history of gynecologic malignancy. She has smoked 1 pack of cigarettes per day for 15 years and drinks several glasses of wine daily. She has not received her HPV vaccination. Her blood pressure is 120/70, heart rate is 71, respiratory rate is 14, temperature is 98.1. A speculum examination shows a nulliparous cervix in the mid-plane of the vaginal wall with red discoloration, approximately 1 into 2 centimeters in diameter. Bimanual examination reveals no pathological changes. A pap smear is shown in the exhibit. Gene coding for which of the following proteins is most likely to be mutated in the affected cells? So is it EGFR, P53, MYC, BTK, or CSRC? Straightforward case of cervical cancer caused by HPV. Okay, now why this question is important is because you need to know the pathogenesis. You need to know how HPV is causing this, why HPV predisposes patients to cancer. So let us look at this diagram. Okay, so the oncogenic potential of HPV can be related to products of 2 viral genes. So HPV has 2 genes, the E6 and the E7, which are responsible for pro-oncogenic activities of HPV. So let's talk about E6 first. The E6 protein binds to and mediates the degradation of P53. It also stimulates the expression of something called the TERT, which is basically this gene helps in proliferation of the cells. Okay, so which contributes to immortalization of cells or proliferation of cells. So the E6 so HPV, there are low risk variants and there are high risk variants. So E6 from high risk HPV types has a higher affinity for P53, and this is what likely contributes to the oncogenesis. Okay, so E6, you need to remember, inhibits P53. And activities of E7, the E7 protein has effects which complement those of E6. And so basically what it does is it tries to increase the cell proliferation by speeding cells through the G1 to S cell cycle checkpoints. So it binds to the RB protein and it displaces the E2F transcription factors that are basically, it binds. You don't need to know the details of that. It binds to the RB and it promotes progression through the cell cycle, and thus it promotes proliferation. And just like with E6, E7 from high risk HPV types have a higher affinity for RB than from low risk, and that is how it carries out uncontrolled proliferation of cells. So that is exactly what you will see they're saying here. The pap smear shows intensely stained eosinophilic cells with prominent nucleoli and mitotic activity. The smear is suggestive of cervical adenocarcinoma. More than 90% of cervical cancers are associated with HPV. HPV is oncogenic. It is non-enveloped. It is a double-stranded DNA virus capable of integrating its DNA into the host genome. And such integration affects the proto-oncogenes, and malignant cell transformation and proliferation can occur. So there are more than 100 types, but these 16 and 18 are the ones you need to know because they're the high-risk variants which cause cervical cancer. Now, okay, so HPV has a number of early and late genes transcribed early and late in the virus cycle. Okay, you don't need to know that. You don't need to know the difference between early and late, but basically E6 and E7 are the 2 genes responsible for carcinogenesis. So cervical cancer, which is HPV 16 and 18 positive, if they will have the E6 and E7 proteins. E6 is responsible for inhibiting the wild type P53, and E7, the RB tumor suppressor gene, it gets inactivated. Okay, so the main function of RB is to stop or slow down or regulate progression from the G1 to S phases of the cell cycle. So when we inhibit RB, there is uncontrolled progression through the cell cycle, and that is what causes defective cells to get through. Okay, so E7 inhibits the RB protein, which inhibits proteins promoting cell division. E6 inactivates P53, which is responsible for cell cycle regulation, induction of apoptosis, and promotion of DNA repair. So that is what you need to know in this question. Some of the other important markers as well. So EGFR mutations are associated with different types of cancer like breast, head and neck, lung, but not with cervical cancer. The MYC is associated with it's the gynecological cancer, but the strongest association is ovarian cancer, but not cervical cancer. BTK is an important regulator of lymphocytic proliferation, and SRC is associated with breast, prostate, and colon. So E6, E7, E6 with P53, E7 with RB, these are very high yield points to remember.
Thanks for watching. Please give a thumbs up and subscribe for more.